Good evening, ladies and gentlemen. Thank you for the opportunity to share my research journey and my current perspectives of the work that has been done, its implications, and my reflections looking back at the 40 years of research that I've carried out. 40 years is hard to believe, right? Yeah, it's a long time. So I like to start the lecture, as usual, like in my classes, with a trivia. So anyone that can answer this? What is the most abundant organism in the world? Any answer? Bacteria? Virus. Yeah, it's a virus, of course. And it's a virus that was discovered in 2013, and the virus feeds on the bacteria, okay, which is a marine bacteria called SAR-11. So what are viruses? They are infectious agents that typically consist of a protein code that contains this is the protein code, that contains the hereditary material, which can be either DNA or RNA. And they come in all types, shapes, and sizes. Maybe you like to find one who is your pet virus. You know, I like this. It's a bacteriophage. It looks like the lunar landing module. Some argue that viruses are not organisms. This is because they are obligate parasites, which means they need a host, like me or you, or your pets or your plants. Those are the hosts to sustain themselves, and reproduce more progeny viruses. But of course, we often associate the viruses with HIV, polio, measles, influenza H5N1, hand, foot, and mouth, herpes, Zika, is quite terrifying at the moment, hepatitis B, Nipah, SARS, dengue, and of course, you may not have heard it, but it's NDV as well. The climatic changes that we are facing, coupled with rapid urbanization and mobilization, provide the perfect storm for a host of emerging pandemic diseases, such as the recent Ebola, Zika, SARS outbreaks, and closer to home, the dengue epidemic and the chikungunya outbreak. But are viruses our biggest problem? No. Statistics released by GlobalCan a couple of months ago is nothing short than alarming. The global cancer burden is estimated to have risen to 18.1 million new cases and 9.6 million deaths in 2018. And it is estimated that cancer will at some time be the biggest cause of premature mortality in most countries. So, can you expect this map? This is a map, yeah, that shows the blue is the ones with a lot of cancer. Can you expect to see this map turn completely blue in the future? The lifetime risk of cancer is 1 in 2 for Canadians, British, and the Australians, and the Americans 1 in 3. In Malaysia, the lifetime risk for cancer by the age of 75 is 1 in 10 in men and 1 in 9 for women. Cancer is common in our community. I'm sure many of you would know at least one person who has got cancer, right? So, and it will get more common, matching that of the Western world. Hence, the war on cancer, initiated by the College of Physicians Malaysia, under the leadership of my husband, in collaboration, he's here, in collaboration with a host of other organizations and individuals, including cancer-focused organizations, was launched by none other than the Deputy Prime Minister herself in Malaysia recently. This is a war that we have no choice but to win. The reason why this war on cancer is extremely pertinent is best exemplified by the limitations of the mainstream therapeutic modalities, such as surgery, chemotherapy, and radiotherapy, especially for the late stages of cancer. Even in the developed world, such as the UK, overall 5-year survival is about 50%, with great variance between cancers. Survival rates can range between 1% to 98%. The ability of cancer cells to develop resistance to treatment is now well organized and of great concern. I'm sure that you are all well aware that some viruses can cause certain types of cancers. For example, the hepatitis virus causes liver cancer. These two are different viruses. The human papilloma virus causes cervical cancer, and the Epstein-Barr virus causes nasopharyngeal carcinoma. There are other viruses. The mechanisms by which these viruses cause cancer are either by integrating into the human DNA, thus causing mutations that result in the activation of oncogenes and inactivation of tumor suppressors that would otherwise protect the integrity of the DNA. These viruses are also able to elicit an epigenetic effect that results in genes switching off or switching on that transforms the normal cells to cancer cells. However, today, my story is how these bad guys can come good in our war on cancer. Sometimes, ladies and gentlemen, to win a war against one's enemy, we may have to sleep with other enemies. Newer modalities that add to our therapeutic armamentarium provides us with confidence that our war on cancer, especially against resistant cancers, may be won. Viral therapy is emerging as one of the newer modalities. Viral therapy exploits the potential of naturally occurring and engineered oncolytic viruses to selectively replicate and cause cytotoxicity to tumor cells without affecting healthy normal cells. The decision by the FDA in 2015 to approve Acknowledgements as the first oncolytic viral therapy in humans is a major landmark. Other countries such as Latvia, Russia, and China had introduced their own variant viruses earlier on. This is Acknowledgements, which has been approved for the treatment of late cancer stage melanoma. But our story starts here. It starts with the humble chicken. Now, we all love chicken. I hope, I love my KFC as well, you know. Yeah, but this is Newcastle disease virus. It is an avian paramyxovirus serotype 1 that infects all types of birds. And there are 3 pathotypes: the lentogenic, the mesogenic, and the vallogenic. The lentogenic is avirulent, and the vallogenic is very, very virulent. While NDV is not generally virulent against humans, where it causes only mild flu-like symptoms, conjunctivitis, and laryngitis. The poor chickens are not that lucky. You can see here, all of them have died because of NDV. Infection by NDV may result to 100% mortality, resulting in significant trade barriers and severe economic loss. My love story also begins with NDV about more than 30 years ago at Newcastle upon Tyne. It has been around 30 years since I've worked on characterizing the various genes and protein domains of NDV when I was doing my postdoctoral research at the university there. The NDV genome is a single-stranded, non-segmented RNA of about 15.2 nucleotides, consisting of 7 genes which encode for specific proteins. So we did sequencing in all the genes on the genome. It's not big, it's small. That's why I use viruses, because it's easy to work. The virus is pleomorphic, which means that the shape and the size varies from 100 to 500 nanometers in diameter. It changes all the time. The virus is enveloped with a helical nucleocapsid. So, a Malaysian NDV strain, AF2240, was isolated in the 1960s by the Vet Research Institute. And this strain is highly heat resistant and very, very virulent. It's often used as the challenge strain for our vaccination programs. My colleagues and I at UPM have been exploring the potential translation opportunities of Newcastle disease virus. This includes the development of oral vaccines for poultry. We characterize the nucleocapsid protein, and this is the nucleocapsid protein that looks like stacked donuts, but in a helical structure. Aren't you getting hungry? Yeah, thinking of donuts? We didn't have donuts outside just now. Some of the work we have done with the NP protein are to delete portions of the NP protein from both sides, the N-terminal end as well as the C-terminal ends. We did this to understand the effect of these mutants on the protein structure. The different micrographs clearly indicate the different shapes. These are all N proteins, and you can see the different shapes that we can observe when different portions of the protein have been deleted. So we also found that the NP protein can be used as a carrier protein for foreign proteins of interest by creating a fusion. We can fuse the NP protein with another peptide to form a fusion nucleocapsid. Yeah. And we can use this in diagnostic kits, also in maybe novel therapeutic biologics. This schematic diagram describes the majority of work in using our understanding of NDV biology to understand pathogenesis, as well as to develop various kinds of applications. In recent years, though, my attention has been diverted to the oncolytic capacity of Newcastle disease virus. NDV has several advantages that include the ability to replicate selectively in tumors, up to 10,000 times more efficient as compared to the normal healthy cells. The ability to directly kill the cancer cells that it infects, and the ability to elicit a systemic immune response against cancer. And the different pathotypes display a spectrum of oncolytic activities. So, how does NDV kill the tumors? By direct killing, it activates the intrinsic and extrinsic apoptotic pathways, as well as the endoplasmic reticulum pathway, stress pathway, and the specific tyrosine kinase receptor pathways. By indirect killing through the activation of both the innate and adaptive immune responses. So let's just follow this video. I've got this video to show how NDV, how do we get NDV-mediated oncolysis. The cell receptor binding membrane, yeah? So how do I do it? Okay, I know. Okay, the cell receptor binding, the virus fuses into the cell, and the ribonucleoprotein is released with genome translation into viral proteins, as well as the replication of the genome. And then the, let's see now slowly, the HNF protein translocates to the surface of the membrane, and the M protein goes to the mitochondria, triggering apoptosis. And then there is ribonucleoprotein assembly and membrane translocation to the surface. There's viral assembly and release. And then the cell dies. A few clinical trials are underway with promising results for NDV. However, many of the clinical trials were conducted in patients who either had advanced disease or no other therapeutic option. Nevertheless, the general consensus has been that NDV is safe, and some patients demonstrate positive responses. So we have here clinical trials that's been done by NDV. This is by NDV oncolysate, as well as the autologous tumor cell vaccines. For example, you see here, this is NDV therapy in breast cancer. They treated with NDV after surgery and showed dramatic post-operative survival rates as compared to patients without NDV therapy. In another trial, a man with tonsillar carcinoma showed dramatic complete response after 3 months post-treatment. You can see here the tumor before the treatment and the disappearance of the tumor. Same here in this diagram here, tumor and the disappearance of the tumor after 3 months. In another trial, in a recurrent glioblastoma multiforme, where patients have very poor prognosis, the scans show the shrinking tumor. You can see here, that's the tumor. It's shrunk after 25, 30 weeks from the start of virotherapy. These data, ladies and gentlemen, are indeed very promising. So my group has further been trying to characterize the strain AF2240. This includes investigating its potential cancer targeting abilities, as well as its immunomodulatory effects. As a result, my colleagues and I have characterized the strain AF2240 mediated oncolysis through a whole range of tumor types. It's not only my group, but the various groups in UPM. we have shown that NDV-mediated oncolysis occurs through apoptosis and have developed and have identified clues towards its mechanism of interaction. Here you can see that the cells, after treatment with NDV, the cells all die. Yeah? The cells which are not treated with NDV survives. This image here shows that NDV induces cell death in cancer cells through apoptosis, as evident by this electron micrograph here. You can see cell blabbing, and you can also see here the red color shows cell death after staining. We still don't know the exact mechanism of action in which the virus selectively targets. As evident from the clinical trials, although some individuals respond very well, there is great variability across patients, across cancer cells. One of the reasons for the variation in efficacy is the development of persistency of infection. Let me explain to you what persistency of infection is. We all anticipate that when these oncolytic viruses target a tumor, this is how they target the tumor, the viruses infect the cancer cells and the cancer cells lyse. That's the ideal scenario. However, more often than not, the viruses infect the cancer cells, but some cancer cells survive that infection. They remain to be alive. That is what we call persistency of infection. These persistently infected cells are potentially like time bombs. That may acquire the ability to rapidly expand into resistant tumors. And you don't want that. You don't want that to happen at all. In this slide, we have two types of bladder cancer cells. Yeah? And that is the sensitive cells, and these are the resistant cells. And this is before infection. You can see that the sensitive cells all die after viral infection, but the resistant cells survive. So our main aim now is to try and make sure that these cells which are surviving are killed. That's what we really aim for. So how do we kill these persistent cells? How do we overcome all these limitations? We can do either by transcriptomic regulation to look at the mechanism of resistance that occur in the persistent cells, or we can do genetic engineering. I'm sure you all have heard of genetic engineering, yeah? So to be able to prove the efficacy and standardize virus production, reverse genetics is increasingly being employed to develop a host of recombinant virus that are armed with various killing protein-producing genes. So there is a lot of work that's been done by various other people around the world looking at this using reverse genetics. But what is reverse genetics? This diagram looks very, very complicated. In our lab, we have developed this reverse genetic system to produce a recombinant Newcastle disease virus strain AF2240. We call this strain RAF. As complex as this may seem, it can be simply put that we have a plasmid vector that harbors the complete cDNA genome of the NDV strain, which allows molecular editing in the lab. This large plasmid, this one here, we have here, okay, will be transfected into a genetically modified cells, which is tailored for recombinant Newcastle disease viruses, named BHK GMO strain. That's somewhere there. Along with this large plasmid, we can also transfect the 3 helper plasmids to facilitate in the formation of the recombinant plasmid genome. Once the full genome is transcribed along with all these various other proteins, we actually get a recombinant Newcastle disease virus. However, we have to propagate the virus in embryonated chicken eggs by harvesting the media of transfection and injecting them into the eggs in order to get sufficient virus quantity for our downstream experiments. Using reverse genetic system, we have developed recombinant Newcastle disease viruses with different types of immunomodulatory genes that can help to enhance the systemic killing. This is especially important in the treatment of patients with micro and distant metastasis. Excuse me. Our current work is about assessing the delivery of these genes of interest and the effect it has on the safety and efficacy profiles. We are looking at how the virus immunostimulates or carry out oncolysis. Transcriptomically, see, I can pronounce that. We have also characterized various genes that are differently regulated between NDV-sensitive and NDV-persistently affected cells. This heat map here, this heat map is a dendrogram that shows that there are selected groups of genes that are upregulated or downregulated in both persistently infected cells as well as in sensitive cells. So these differentially regulated genes can distinguish these cells, and we can do a lot of studies now that we know which genes are upregulated and which genes are downregulated. And we are now currently dissecting these pathways. We are looking at crucial genes associated with cancer cell survival, which is associated with persistency of infection. We are looking at the cascade effect of global gene transcriptional changes, which are depicted through the pathway analysis of dysregulated genes, and multiple interacting signaling pathways, which are associated with the effect of specific microRNAs. We are now exploring ways in which we can modulate the gene expression of crucial genes to reverse the persistency of infection phenotype. We also tested our recombinant virus against the wild-type virus in tumor-challenged mice. RAF and wild-type NDV were injected intratumorally into the mice. This image shows the number of mitotic cells. Yeah? And you can see here that there are more mitotic tumor cells here in the negative control as compared to the recombinant virus. There's only 1, 2 here. Yeah. The RAF-treated mice tumor was the smallest. That's the RAF-treated, the recombinant Newcastle disease virus-treated mouse. The tumor was the smallest, and compared to the negative control and the treatment of the wild-type NDV. This is, of course, really exciting results, and we are currently exploring avenues to improve the RAF with different immunomodulatory genes, but also identifying how we can harness the transcriptomic knowledge to better equip RAF as a cancer vaccine, thus inching our way closer to translating RAF into clinic. So, ladies and gentlemen, the journey continues. However, one of the greatest challenges is not necessarily a scientific one. It is the cultural acceptability of using viruses to treat cancer. No thanks to zombie Hollywood movies like this and disproportionate skepticism that defies logic, which may hamper effective translation of the virus into clinic, and thus the engagement with various stakeholders to prepare them for the eventuality which is necessary. I have been spending so many years trying to find money, and I'm still searching for more money to do my research. So if anyone, some good Samaritan wants to donate, I'm willing to accept. After 40 years of research, I've also come to terms that much of the grand challenges affecting humanity cannot be answered by the scientific enterprise alone. There are many factors that needs to be considered. And one is, of course, the new meaning of going viral in the 21st century. We are not talking about the actual viruses, but something more pathogenic that is threatening the very fabric of humanity. That is, can anybody guess? That is the corruption of knowledge and the corrosion of ethics. The first corruption is the fake news. We heard a lot about fake news during the last general election. Okay? And the rise of alternative information, commonly known as lies. And an MIT study did the study, and they studied on 126,000 posts on Twitter, tweeted by 3.5 million users between 2006 and 2017. The lies are 70% more likely to be retweeted than the truth. Are you all doing that as well? Because the lie seems to be more interesting. Truth takes 6 times longer to be seen than lies. Humans were more likely to spread fake news than bots. So is having anti-fake news laws the solution? The epitome of fake news is the rise of anti-vaccination movement that seems to be able to spread lies so effectively. For example, would you think that Mahatma Gandhi, all of you know Mahatma Gandhi, would you think that he would have said this? Vaccination is a barbarous practice and one of the most fatal of all the delusions current in our time. Conscientious objectors to vaccination should stand alone, if need be, against the whole world in defense of their conviction. Can you believe it? So when we read your WhatsApp or whatever next time, please use your head. The anti-vax movement is giving rise to infections that previously were thought to be effectively eradicated and close to be eradicated. Scientists seem to embrace effective and empathetic communication of science to ensure that facts trump lies. But the problem also lies within us. Perhaps the loss of trust in the scientific enterprise has been the result of hijacking of the original purpose of science. The purpose of science is to improve the quality of life, improve our environment, seek wealth creation, promote peace, and the pursuit of knowledge. There is also a conflict between purpose and consequence. How prepared are we to mitigate unpredicted consequences of the science and technology advances? For example, who could have predicted the impact of social media? Then there is the conflict of interest. Does our science benefit the majority of people or just a few individuals? This is best exemplified by the development of a new cancer drugs that can only be afforded by the rich. So the question is, does science serve scientists or humanity? I'll repeat. Does science serve scientists or humanity? And to compound this problem, there is also growing research misconduct. So we ask ourselves, what is the end game of our research? Is our research relevant and it will serve the community? Or are we doing the research, like me doing recombinant NDV, for instance, for personal glory and fame? Are we prepared to be ready for the various ethical dilemmas associated with these new technologies and experiments? You all have heard about He Jiankui, yeah? So what are your thoughts on his recent announcement on gene editing? Do you approve? Or is it too early to say anything? The reproducibility of work and scientific fraud has resulted in rise of paper retractions. Boon Peng, sorry, I couldn't put in all those paper retractions by one of our universities here. All of these does not serve our scientific enterprise any good. So, ladies and gentlemen, the Global Research Council has encouraged individual countries to come up with their own code on responsible conduct in research. Malaysia has enthusiastically responded to this call and had its own Malaysian Code of Responsible Conduct in Research that has been endorsed by the National Science Council last year. The MCRCR is being adopted by funding organizations, particularly from the Ministry of Education, as well as the Ministry of Energy, Science, Technology, Environment and Climate Change of Malaysia. The Young Scientist Network of the Academy of Sciences Malaysia had taken the responsibility to be the agent to conduct training courses for researchers from various universities and research institutes. For this, the Malaysian society is ever appreciative of the Young Scientist Network. Give them a clap. There are many there. For their enthusiasm and foresight in supporting this effort. So, ladies and gentlemen, you will see that the scientists in Malaysia are capable of achieving substantial scientific accomplishments, and they are capable of self-regulating themselves, as exemplified by this education module and the NCRCR. So we had our training workshop in my lab at UPM. So you can see how happy we were. I think we were happy because it's also the end of the course. Let's admit it. So we have to start at every level of our scientific ecosystem to ensure that RCR is embraced by the whole ecosystem. I would like to thank here my collaborators and students, Ben, Len, this is Howard from the NIH, Professor Latif, he was my mentor. Aini works with me for our chicken work. Raha, I share the lab with her. We do our surface display work. Abi, he's the immunologist. Aziza, Nor Aziza Shafi, and Winston, you're here. Put up your hand, at least, you know. Yeah, there's Rahman, Jahan, Aidin, Eddie, Chui Ling, Xiaowei, Sun Choi, Kavita, Alan, and there are others as well. Sarah, Mokresh, Aline, Kok Song, Jeff, Siu Kong, Irwan, Aaron, and Saila, and the rest. Yeah, Fred and Jivan and the rest of my students there. I think the students are here and they are trying to see whether I can remember the names. I have a lot of more people as well, yeah, from my lab. So special thanks to University Putra Malaysia for providing me with this, this opportunity to do my research, and also for special thanks to my family. My husband is here for his continuing support. I started this trivia. You remember I asked you a question just now? What is the most abundant organism? So I would like to end this lecture also with a trivia. I would like to end with this picture here. Can anyone guess who this is? He's not my grandfather. Okay, John Monash. I mean, it's obvious, right? Yeah. So I want to end today's lecture by thanking once again Monash University Malaysia for giving me this opportunity to deliver this lecture. As many of you are aware, today is 100 years after the end of World War I. In that war, Sir John Monash proved to be a celebrated military commander. After the war, he did many, many, many things, as Maud said just now, yeah, including support for education. He left many legacies, including a university named after him. But I would suggest that his quote, "Equip yourself for life, not solely for your own benefit, but for the benefit of the whole community," is a continuing legacy that is relevant to what I have just shared with you. Thank you all.
Thank you very much. I think you will agree with me that that was a most marvelous lecture. Can we give her another round of applause?
Okay.
Thank you very much, Khatijah. Yes, so Professor Khatijah has covered everything and tried to make even the more complex science palatable and understandable to most of us. And not only did she cover start off with the cancers and the aspects of her work, I think she also built in aspects of community, aspects of ethics, and so on that are very, very important. So it was a very well-rounded lecture. Professor Khatijah will now take comments and questions from the audience. So can I request that you please identify yourself first and your institution or where you're from before you ask the question. And just raise your hands. We've got microphones.
Hello.
There and there, okay, on both ends and in front as well. Yes, please introduce yourself, where you're from, and then give the question to Prof Khatijah.
Prof, Assalamu alaikum. Thank you very much for your excellent presentation on virotherapy. I am from School of Engineering. My name is Ahsanul Kaisar. So I saw that in the in vivo model, you have done the intratumoral injection, right?
Yes.
But my question is, why you did this intratumoral? Why you didn't try for the intravenous? because if you do with the intratumoral, so you can see the tumor, so you can do the intratumoral. But if you cannot see the macrophage or microphage of the cancer cells, so then how can you treat them?
We are also doing intratumor experiments on animal models, but I just showed here the tumor because it's easier for you to observe the tumor on the slide. Yeah? And we are still getting results because the experiments haven't been completed yet.
Hi, Prof. Thank you so much. First of all, you're such an inspiration for all of us. So I have two questions. Number one,
can you introduce yourself? And
I'm sorry, I'm so sorry. I'm Vinod from Jeffrey Chia School of Medicine. So the first part of the question is, did you look at the potency or the potency of killing of tumor cells of the recombinant virus from UPM between different types of tumor? Is there like a blanket mechanism for all? Does it kill the same rate?
No, the viruses, it seems strange that it doesn't kill, it kills different types of tumors at different rates, and sometimes there's no effect at all. So, but our lab is concentrating only on colorectal cancer and bladder cancers. And what we found is that they're quite potent.
Okay. So the second part is, does NDV, or especially recombinant NDV, causes methylation changes? So should I be worried if I want to use it for a glioblastoma cancer, for example?
Well, the thing is, NDV is, it has got no DNA intermediate. So it is pretty stable, and I wouldn't expect it, but then we need to do some testing before we can even say that.
Because we are trying to work with Zika as a anticancer for glioblastoma, and our first sequencing result shows that it causes massive methylation changes in progenitor cells. So that was the question.
Yeah, we didn't look at that.
Okay.
But perhaps you may like to collaborate with me and look at that for me.
Thank you.
I love collaborators, you know. I've got a lot of collaborators.
Sure, Prof. Thank you.
Just raise your hand so the people can get to you with the microphones. If you have a question, yes.
Hi, I'm Avi from Sunway. Thanks for the lecture. And I really like the end bit when you talk about, the larger questions that, perhaps the scientific community is perhaps the problem rather than the solution. So my question to you is, you've rightly spoken about the threat of the irresponsible conduct. So apart from what the Young Scientist Network has done, and as also the MCRCR top-down, right? What are the things that perhaps universities should be thinking about to ensure that researchers are more focused on the purpose of science rather than playing that game?
I think the Deputy Vice Chancellor is here. I wouldn't want to say, but I would like to forget about KPIs. That's the first thing I would say. Remove those KPIs on publications and we'll be all right. But on a serious note, we should really try and understand what are we doing, what is our end game, and what are we doing in the lab, and see how can our results translate into what really matters. And we shouldn't take any shortcuts because our integrity is there, and it's very important that we preserve scientific integrity in our place. Yeah. You could also have like to train our researchers so that they embrace integrity, ethics, and so forth, yeah, rather than policing them.
Any other questions? Maybe I will ask a couple of questions, and thanks again for that brilliant presentation. The first question I have is with regards to the work you presented, and you're quite true that people have a lot of fears about when they think virus and cancer, they don't actually think of it as a therapeutic entity. very little knowledge is known about utilizing viruses as a therapeutic entity. So the first question is, how do we work to assuage those fears? Or that's the first question. And what is your opinion on that? And what are the efforts being made there? And the second question relates to the young people I see in the audience, all these young scientists, young researchers, early career researchers, not too jaded yet. But oftentimes they are very overwhelmed by the limited resources we have in developing countries, yes? Compared to people who are, you know, like in Western countries, where the perception is that they have a lot more resources and they're are able to do maybe a bit more. So where do our young people stand, and what sort of qualities do you think they have that would encourage them to do good science?
Okay. The first question is, that's why I'm here. You know, I'm trying to tell you all about my work and tell you not to fear about using viruses to treat cancer. And I hope you can spread the word round as well, because communication on virotherapy is very, very important, and we should sort of spread the news. And I think many of people fear about using viruses because, as I started in my lecture, the first thing that I spoke of was that the viruses cause a lot of terrible diseases to humankind. So we have that fear. We know about viruses causing terrible diseases, but now we need to change our mindset and look at the other way around, that how can we use viruses to treat cancer. Now, the second one is about doing work. I suggest that you just follow your passion in science. If you follow that, and also you learn to share the labs and the facilities, we will get by. Not only lab and facilities, maybe share your knowledge as well. So we should have regular lab meetings, changing of minds, you know, and discussion and seminars. That would be very good and helpful. Maybe inter-institutional ones as well.
Assalamu alaikum.
Salam.
I'm Ishrat from School of IT from here, and I have just two questions which are not very much related to the technical aspects of this research. One is just general, because I was having some news that, I mean, people have started avoiding chicken eating because of this cancer aspect. So, I mean, how would you suggest us? Because some people, you know, if it is a fake news, so we should be avoiding it or not. And my second question was some slide which was stating that perhaps USA, Canada, and they were having ratio 1 in 2, just like that. While for Malaysia, I think it was 1 in
1
9.
in 9.
Yeah, 1
in 10.
yeah. So this means that what I was thinking is just like the lady was saying, they are the developed countries, and their life expectancy and their everything is, in terms of human indexes, they are quite fine. So what would be the reason for this all? For the developed nations, they have more cancers and than we have here.
Okay, first the chickens. Well, I think we worry about what the chickens that we eat. It's not because of the virus, the chicken virus, because it's a totally different thing. The chicken virus is the one that infects the virus, infects the chicken. Yeah, and that's the virus that we use to treat cancer. But the types of chicken that we eat, we would also wonder what sort of feed that they have, and how are they being, the conditions, the living conditions, you know, and the dirt and all that. And I worry about the mutagenic activities of those chicken feed that we eat. Now, the other one is on... yeah, that's why we should have really more stringent enforcement, checking on the food that we eat. The other question is on, do we expect the rates of cancer to increase, lifetime risk to increase? The population in the West is much older, they live longer, but then we are also catching up. So that's why I worry that maybe one day we will really increase our lifetime risk to as much as maybe less than 5. Yeah, that's a big worry. It's our lifestyle as well. That's the cause of cancer. The other thing is actually smoking. Yeah, please do not smoke. Yeah. I think you're so silly to smoke, you know, whoever's smoking. Yeah, you're just killing yourself. Yeah? It's a death wish, yeah. But I don't know. I don't know who is here. I hope you are not smoking. You're not a smoker? Yeah? No, that's good. That's good.
And I like to sort of to continue to this and to be somewhere relevant. Earlier this year, I think we had Sir Boris Lizak, former Vice Chancellor of Cambridge University, who is a cancer expert in Monash University and maybe visited other universities as well. And he was telling us that the 3 biggest risks for cancer would be, number 1, smoking, number 2, obesity, and number 3, old age. So try not to get too old.
But Maud, there's no way we can grow old, you know. I mean, that's so idealistic, yeah.
I know. It was just, so we thought, okay, right. Yes, Prof Mahendran.
Can I ask a question? I mean,
yes.
If you see in most of the OECD countries or developed countries, you've been to many places, there's a lot of fantastic science that's happening, very similar to the work that you're doing, a lot of good fundamental research, but also there is a lot of support for translational research where some of the lab base is translated into products and services and vaccines and a whole range of things that are coming out. But one of the challenge in many developing countries, and I think not just peculiar to Malaysia, is that you've got some fantastic work happening in UPM and other places, but somehow you don't see many of those fundamental research that comes out into vaccines and things that, you know, that become what we call translational impact. You see that in Cuba, some of the work that's coming out of Cuba. But why is it that it is not coming out both nationally and globally? Is there something that as a scientist you've been at different levels, you know, at the university, at the ministry level? What's missing in our ecosystem that doesn't connect the fundamental scientists to industry, or are industries becoming too dependent, or are we dependent on more developed countries?
Yeah, the question, the industries, they are supporting us. For instance, in my work, I have got an MOU with one of the vaccine producing companies in Malaysia, which produces vaccine for chickens. Yeah, but they are very interested in working with me for this human cancer vaccine. However, they are not willing to put that money. Yeah, they are always relying on government support. This is our private companies here, but they still rely on government support. We don't have sufficient funds, and it's also very difficult to try and convince the donors, the two main ministries that provides funding for research. It is quite hard to get them to understand the whole ecosystem of from ideas right up to clinics or to the business of in our research. And also the reiterations that we have, you know, sometimes you have to go back and do more research. The policy makers, they don't understand. I was a policy maker too, you know, I was in MOSTI for 5 years, and I knew how difficult it was to try and get the various other non-scientists to understand how we carried out science, what is really needed to push science further. And also, we don't have enough, the base is too little, yeah? We need a broader scientific base here to support our science.
Assalamualaikum, Datin. I think you know my name.
Yeah.
I'm a friend of this wonderful couple. My name is Dato' Rahim Kamel.
Excellent.
You raised questions of funding, and I would like to ask you a trivia.
Oh, you are asking me a trivia now?
Yeah.
Okay.
It's my turn to ask. What was the virus that was missing from the list that you put up in the slide?
Oh, I couldn't recall.
One MDB.
SARS?
One MDB.
One MDB. Okay. Spot on.
Are there any other questions or comments from the audience? On this side, people have been quite quiet. Yes, there's a gentleman at the end.
My name is Tan. I'm retired. I was an engineer before. But coming back to your funding, I mean, funding is one of the main issues in every organizations. And in your particular case, on cancer, there is also competition among various research groups. For example, immunotherapy and viral therapy and so on, And you say that your viral therapy don't get much funding. Why is that so? Because have you not marketing yourself right, or your branding is not right, and maybe other people are doing better than you. Not in terms of research, maybe they got better marketing people and so on. Maybe you should look at, maybe you want funding, do a better marketing per person. So I do not know. But obviously, every company, either private or public or everywhere, they don't have enough funding. Funding will always not be enough. And that's the reality of research and even in our businesses. So I have no answer, but I just throw in some questions. Maybe your branding may not be right. Maybe you are doing everything good. Your marketing may not be right. I don't know, but I just throw some questions. Thank you.
Thank you. Well, we do have funding, but there's still always insufficient because funding for our research, we are looking at millions. And so what we feel is that we still got a lot more to do before we go for our first trial, clinical phase 1 trial, preclinical trials. But we are looking around for partners and collaborators to work with us to do that first in man that we really want to. That will cost quite a bit because we have got to produce the virus in CGMP, and we don't have any CGMP at all in Malaysia for viruses. Okay, clinical good manufacturing practice. And so we need, even just to make the virus to put in humans, we need a special place to put it, and we don't have that in Malaysia. So that's why we have got, I like your idea of marketing. Yeah, we need to market it, but we have to be very careful of ethics, and also because our results, many of them are still under review for patents. So we're quite careful in what we tell the public about where we are.
Any other comments? Yes, I've got a question, Khatijah. I mean, we read so much about, UREXS, for example, these huge grants that one gets from the Wellcome Trust, from the Gates Foundation, and things like that are, millions and millions, and sometimes even $1 billion US dollars. And these are projects that are ongoing on an international basis for periods of time. How do you think that this particular area of research can be positioned to actually capture a portion of that funding in terms of international collaboration?
We can. At the moment, I'm having a funding with under the Newton Nkrumah Fund with Oxford University. We are looking at working on the Newcastle disease virus. Yeah. So from there, I think it's a stepping stone for us to work with other people in the West. But we have to collaborate with them to get those funding, those huge funds.
Assalamualaikum again. I was just, I'm quite lay person on this, my question. I was listening about like Congo virus, and now this new term, again specific to some location like Newcastle. It seems like place in England. Virus name is originated from some location. So I suppose this virus came from there.
Yes.
Yeah, particularly. So why we do focusing here when problem is mainly somewhere in England? So we are subjects mainly over there. I feel like that.
The Newcastle disease virus was first isolated not only at Newcastle upon Tyne, where it's got its name, but also in Java, which is just around the corner. And because it infects nearly all types of birds, and the birds migrate from the north to the south, you know, all the migration. And so we worry about how endemic the virus is in tropical countries like Malaysia. So it is not about the problem of the West, but it's actually the problem here. Over there, they are able to contain the virus. In fact, it's free from virus, Newcastle disease virus, because of good vaccination programs. Over here, we have good vaccination programs, but sometimes the farmers get to be complacent because, well, their chickens are alive, so why do they want to waste money to inject some more, vaccinate some more birds, yeah? So we do have some problems sometimes.
Yes, we should be collaborating.
Any other question? Maybe one last question? Yes, this gentleman.
Hello, I'm Shahir from Panasonic. One of the biggest changes in treating cancer is the ever-changing nature of tumors. Researchers in the Institute of Cancer Research in London and Institute of Edinburgh, they use a new technique called revolver or evolution. Repeated evolution of cancer, which picks up the patterns in DNA mutation in cancer and uses the information to forecast future genetic changes. My question is, how do you adopt or use artificial intelligence or AI in furthering your research?
I won't be able to answer that, but we use AI, for instance, AI for our transcriptomic analysis, our sequencing analysis, but maybe the machine will be much more intelligent and just pick up all those answers that we are looking for, those genes and all that are affecting our persistency of infection and so forth. Yeah.
Yes, of course.
So, I've obviously had the privilege of knowing you and your family this last, maybe almost a decade now. My question is, I think relative to a lot of us, right? I must say I'm really envious. It seems that you have it all, right? They say every successful woman, you have a strong, solid, supportive husband. It seems quite obvious you have that.
I think I'm the supportive one.
Oh, really? Okay. Oh, no, I'm not sure what I've started now. But what I want to say is, like, how do you balance, all of that, yet championing your scientific purpose as well as championing your own, family and moving it? Like, what's your secret?
I think it's just the passion and the love for science. I think both of us love our job. We even have the office in our room. That's our secret, actually. Yeah. But we do enjoy our research, and our children are also understanding. So that's our secret. We also love the same food. You can see our sizes.
Okay, so I think on that note, thank you very much, Professor Khatijah, for answering all those questions. Thank you to the audience for listening and participating. And now it gives me great pleasure to invite Professor Mahindran Nair, our Deputy President for Research, Monash University, Malaysia, to give you a token of our appreciation.